Yeah, so I really appreciate the invitation and you guys and Lady continuing to put up this excellent seminar series. I'm proud and humbled to be a part of it. So I'll go ahead and begin by saying the unnecessary. I'm probably preaching to the converted in claiming that membranes are amazing in this crowd, but I really think they are. And from my point of view as sort of a biophysicist, I like to just emphasize some really remarkable things that are inherent to living membranes that are almost kind of unique and unmatched in nature. So thinking about them as fluids at the molecular level is sort of obvious and trivial, but these same materials don't really behave like fluids at the macroscopic level. They behave as robust elastic sheets capable of sustaining sort of large deformations and stresses, as you see here for an isolated plasma membrane vesicle being stretched and pulled by tweezers and pipettes and such. And another interesting physical chemical feature of membranes, zooming back into the molecular scale, some of the gradients, chemical and physical, are as I mentioned, sort of practically unmatched elsewhere in nature, with the chemical environment transitioning from completely aqueous to almost completely apolar within the length scale of really just a few angstroms. And the same is true for physical forces. These head groups are pulling in with hundreds of bars of tension, and those are directly opposed and countered by hundreds of bars of pressure here in the hydrophobic core. But membranes aren't just cool and beautiful, they're also super important. And I think the easiest way to illustrate that is to simply point out that something like a third of the mammalian proteome encodes for transmembrane proteins that are stuck in membranes for their entire lifetime. And that, of course, doesn't even account for the hundreds of proteins that associate to various degrees of stability with membranes via protein-lipid interactions, amphipathic helices, post-translational lipid modifications, or just direct interactions with other membrane proteins. So I think it's probably a pretty fair and safe estimate that something like half of all bioactivity occurs at or very close to a membrane. But of course, these membranes are not just the host for all this activity, they're also its guides. And what I mean by that is that in almost every context that it's been studied, the composition and physical properties of the matrix that hosts these proteins changes their activity. So from ligand binding by membrane receptors to transport activity, ion channel open probability, signal transduction through the membranes, and really just generally membrane protein oligomerization, conformation, and function are regulated by membrane composition and properties. And at the same time that these insights have been gained, we're starting to learn that living membranes and mammalian membranes in particular are sort of more complex, more diverse, and more plastic than I think, certainly than I appreciated when I first got into this field. And the way that we've been learning this is riding the wave of this fantastic technology, which is called lipidomics, but really is just mass spec, and lipids are really kind of optimally designed for mass spec for quantification and identification of thousands of lipids with extremely high sensitivity and practically comprehensive coverage. And so just to give an example, a few years ago, we published this paper measuring how the plasma membranes of, excuse me, human mesenchymal stem cells change as these cells differentiate into various functional and kind of different morphological lineages. So in particular, bone cells and fat cells, adipocytes and osteocytes. And so we quantified about 1,000 different lipid species in each sample, in each plasma membrane, and saw that really there are clear cell type specificities to the plasma membrane composition in these three very closely related cell types, and that these can be tuned in a way that I'll show you in just a minute in order to functionally tune cellular differentiation. So obviously, if these three sister cells are so different in their composition, cells from different tissue types must be even more different, and that's also clearly true. So here, just comparing three different ones, both with respect to differences in the hydrophilic head groups and the hydrophobic acyl chains. The details here aren't important. What I emphasize is simply the diversity that is accessible in any given mammalian plasma membrane. And even what we call here the neuronal plasma membrane composition is also quite malleable. And this is shown by isolating neuronal plasma membranes, actually as synaptosomes for the aficionados, and measuring how they change during the first few weeks of postnatal development in a rodent. And what basically, again, to reemphasize this dramatic changes in overall lipid composition, both in the hydrophilic and hydrophobic regions during development. And finally, I mentioned this point about plasticity. We found that the composition and the resulting biophysical properties of plasma membranes are remarkably susceptible to external inputs. In particular, one really interesting one is from the diet, because for dietary fats, it actually is true that you are what you eat. Dietary lipid components are incorporated in toto and directly into cell membranes in vivo. And just as an example here, when mice, either mice cells in vitro or the animals themselves, are fed with an omega-3 polyunsaturated fatty acid, like the dominant component of fish oil, this fatty acid, which the mice themselves don't make and basically never see, gets incorporated to a dramatic extent into mammalian membrane lipids, plasma membrane. So general focus of our lab is to try to understand these kind of connections, quantify the detailed lipidomes of specific cell membranes, understand how these lipidomes give rise to the various biophysical properties that are accessible in the membrane, and then how these two combine to ultimately determine functional phenotypes. And within that broad context, I'm going to tell you a quick story today about a paper that came out just a couple of months ago on the molecular and structural asymmetry of the mammalian plasma membrane. So I guess for this group, these cartoons need no introduction or explanation. All of you probably know that the mammalian plasma membrane is highly asymmetric with respect to the lipid head group composition, with zwitterionic lipids enriched on the outer leaflet and anionic lipids almost exclusively retained on the inner leaflet. And of course, glycolipids that many of you are interested in are exclusively outer lipid. So this plasma membrane asymmetry, the function of it has largely been considered in the context, not actually of the steady state asymmetry, but what happens when it's lost. That is when anionic lipids like phosphatidylserine are exposed on the cell surface, on the outer leaflet where they should not be, or usually are not, and a variety of physiological consequences result from that, most notably apoptosis, but many, many others. That said, I think it's important to point out that, again, these are the functions of the loss of membrane asymmetry. And I think it's still not entirely clear why cells spend all this effort to pump lipids against their concentration gradients and maintain this steady state distribution. So just a very quick step backwards into the history of how cartoons like these came about. The measurements are actually quite simple to understand and easy to do. We isolate human red blood cells, and red blood cells are optimal for this because they only have a plasma membrane. They don't have a bunch of internal organelles to confuse your analysis. And then these red blood cells are treated with head group specific lipid enzymes, lipid cleaving enzymes. So lipases, sphingomyelinase, for example, phospholipase A, phospholipase D. And these lipases will clip one head group at a time. And then you can analyze what's left over, and by that infer that that must have been on the inner leaflet, inaccessible to this externally applied enzyme. And you do that one at a time for various lipid head groups, and that's how you establish data that looks like this. And all of these graphs that you see in textbooks and all over the internet derive from basically this one paper from almost 50 years ago, which did some beautiful biochemistry exactly in this way. But I think it's important to point out that the legend to this original figure very clearly specifies that this is not real data, and that's how you can tell there's no data points or error bars anywhere. This is something like a cartoon that was established from really one set of experiments. And so we thought that maybe it would be interesting to revisit these conclusions, particularly in light of the obvious fact that there are really thousands of different lipids that we know exist, not just these 4 groups, and to sort of analyze how they are distributed between the 2 leaflets of the plasma membrane. So we took basically this exact same approach, but now combined it with sort of modern quantitative lipidomics to measure the species asymmetry, species level lipid asymmetry. And so this is sort of what data looks like after a bit of processing. Kind of think of these as lipidomic fingerprints that can be compared between various samples. It's sort of hard to derive anything particularly meaningful from complex data like this. So the self-similar lipids can be basically clustered together. You know, you can imagine that these 2 polyunsaturated PC species are similar enough that they can be treated as a group. And so with a little bit of subjective clustering, this is what the distribution of lipid compositions look like in the inner and outer leaflet of the human plasma membrane in red blood cells. So just with, you know, about 6 or 7 different lipid species, you can really capture all the relevant diversity of the outer leaflet. The inner leaflet requires a bit more because there's head group diversity, a little bit more head group diversity. But again, just with about 10 or 12 different lipids, you can capture all the relevant stuff. And you can find the full lipidomes here. So what's sort of the main thing that I learned from looking at this kind of interesting complete data? Well, the first thing that I learned is that Dutch people are very good at biochemistry, and they shouldn't be messed with even if their results are 50 years old. Because when we compared our resulting head group distributions to those ones that were published in the early 70s, they were really remarkably similar. And so my face after 2.5 years and $20,000 of lipidomics was sort of like, I can't believe that. They got everything right. So annoying. But of course, we have access to more data than they had, because lipidomics gives us all of the structural information in these lipids. And so, you know, for example, the number of double bonds in any given acyl chain, or the connections between the acyl chains and the head group. And there we did find something interesting, though not entirely shocking, which was a dramatic asymmetry in the overall lipid unsaturation between the outer and inner leaflet. So this is what you're seeing here. The outer leaflet was sort of, the major lipids were fully saturated. These are the sphingolipids that many of you know about. Whereas the inner leaflet was much more unsaturated, with the majority, indeed, of phospholipids in there being polyunsaturated. Oh, I should mention, because this is a sphingolipid interested crowd, we actually did not directly, using this enzyme way, measure the glycolipid asymmetry. We basically just assumed that all the glycolipids are on the outside, and together the major glycolipids in the red blood cell account for something close to 1% of all two-tail lipids, that is non-cholesterol lipids, in the outer leaflet of these plasma membranes. Okay, so now coming back to this acyl chain asymmetry, those glycolipids would be mostly here, largely saturated, and they in total help to make the outer leaflet more than half as unsaturated as the average inner leaflet phospholipid. So for us as kind of biophysically minded types, this brought up a really, I think, obvious question, which is, now knowing that these 2 leaflets are highly asymmetric with respect to their acyl chain saturation, what would that mean for their properties of these 2 leaflets, right? So in the trivial biophysical prediction would be that a leaflet very rich in saturated acyl chains would be tightly packed, highly ordered, perhaps less fluid, whereas a loosely packed inner leaflet might be the opposite of all of those things. But that, of course, doesn't account for the possibility of direct biophysical coupling between these leaflets. So I think it was important to try to ask this question, is there biophysical asymmetry in a living plasma membrane? So the first way that we approach that is with a computational model. We built membranes that are representative of these pie charts that I showed you earlier. So these are all atom simulations containing many thousands of lipids and trying to, as best as possible, completely recapitulate all of this diversity, both in the head and acyl chains. And these were simulations done by my friend at Lyman. And the first thing we observed is exactly in line with our prediction, that a saturated lipid-rich outer leaflet is much more ordered, approximately twice as ordered as the inner leaflet. So all the acyl chains are much more stretched out. And also that the inner leaflet is much more fluid, with any given phospholipid diffusing about twice as fast in the inner leaflet compared to the outer leaflet. And again, consistent with all of that, the outer leaflet was more tightly packed with a smaller area per lipid. And another interesting thing is if you look at hydrophobic defects, right? So now if you look down on a membrane from the top, mostly you see head groups, but you do see areas where the hydrophobic acyl chains are kind of accessible to the water. They're small, they're transient, but they exist. And the frequency and size of those defects was much smaller in the outer leaflet because the lipid head groups were more tightly packed. So essentially, this is the kind of computational prediction that comes directly from our lipidomics, that there is indeed biophysical asymmetry in the plasma membrane, with the outer leaflet being more ordered, more tightly packed, and the inner leaflet being the opposite. And so we basically wanted to test this prediction directly, experimentally. And so to do that, we needed a probe that could do two things at once. The first thing it would have to do is be sensitive to this property, something about the biophysical parameters, order, packing, fluidity. And the second thing it would need to be able to do is to do it one leaflet at a time. So the first part of that is easy. There's a variety of fluorescent probes that are sensitive to membrane packing. Lordan is probably the most famous one, but another one here is shown is called DI4. It has an emission spectrum that sort of red shifts in more fluid membranes, blue shifts in more tightly packed membranes. But also its fluorescence emission lifetime, that is how fast it releases photons, is also highly dependent on membrane packing, with a much longer lifetime in ordered versus disordered membranes. And that can be directly imaged by fluorescence lifetime imaging, so FLIM. So here's a phase-separated giant unilamellar vesicle made of DPPC, so saturated PC, unsaturated PC, and cholesterol, which is known to phase separate into coexisting ordered and disordered phases. And here you can see this really dramatic divergence of lifetimes that can be directly measured in these vesicles. So, okay, now we have a probe that's sensitive to biophysical properties, but how do we do it one leaflet at a time? Well, it turns out that this DI4 is really perfect for that, because it has this quote unquote head group moiety that is highly charged. And these charges absolutely, or rather almost completely, prevent it from flipping across the hydrophobic bilayer core. And we sort of validated that in a variety of control experiments that I don't have time to get into, but you're just gonna have to trust me for now that it really doesn't flip on experimentally relevant time scales. The half times are on the order of hours for flipping. So then staining the outer leaflet in cells is really easy. We just add the dye to the kind of bathing medium, and it incorporates into the outer leaflet and stays there. Now for the inner leaflet, it's a little less easy, but also doable, because this dye can simply be micro-injected into the cytoplasm, where it will then incorporate into the cytoplasmic leaflet of the plasma membrane and kind of all the other internal membranes, the ER, the endosomes, etc. So now we have the same exact probe in one or the other leaflet, and then we can directly compare the readout from that probe. And this is what the data looks like. So this is this dye-4 micro-injected into a kind of a model mast cell. And what you're seeing encoded in the color is the lifetime of dye-4, with warmer colors representing higher lifetimes and therefore representing tighter lipid packing, more ordered membranes. And unsurprisingly, we see that the plasma membrane here looks kind of green, whereas internal membranes look kind of blue. So the plasma membrane is more tightly packed. This is exactly what we would have expected, although I should note that we kind of presume that this is inner leaflet only, right? Not overall membranes, which is that cytoplasmic leaflet of the plasma membrane is more tightly packed than the cytoplasmic leaflets of internal membranes. So now what happens if we try to stain both leaflets? Well, we can take this very same cell and add the dye from the outside. Now what I hope you can see is that the internal membranes don't change color, which makes sense if the dye is not penetrating the cell, but the plasma membrane does. So whereas the inner leaflet was reporting a relatively low lifetime, now together with the outer leaflet, we get a much warmer color and a much higher lifetime in this clear shift. And now we can confirm that by taking some new cells that weren't micro-injected with the dye, staining them only from the outside where we only see the plasma membrane, and we get sort of a warmer color still representative of higher lifetime and tighter packing. So that's sort of fully consistent with that computational prediction that the outer leaflet seems to be more tightly packed than the inner leaflet of the plasma membrane. And we can do that across cell types. We've done it with 3 different ones now. That's not on this graph. And consistently, we see that the exoplasmic leaflet is much more tightly packed than the cytoplasmic leaflet. And just to compare, to give some sort of benchmark for what these lifetime values represent, the outer leaflet looks almost as packed as a pure lipid liquid ordered phase, whereas the inner leaflet is approximately intermediate between the ordered and disordered phases in synthetic model membranes. Okay, so that's the biophysical asymmetry of the plasma membrane covered. So then we started getting interested into thinking about how we can measure asymmetry in internal membranes. And the low-hanging fruit here was simply endosomes, because we could stain those by first staining the plasma membrane and then just waiting, right? Letting the cell sit. The dye would get passively or actively endocytosed and accumulate in intracellular structures. And to stain those intracellular structures that came from the membrane, we bathe the cell in fluorescent dextran, which also gets taken up and accumulates in these compartments. And that's what you're seeing here. And the membrane of these compartments is stained by the dye-4, which was originally on the outer leaflet of the plasma membrane, right? So these are the cells stained only from the outside. And what I hope you can see actually right from the color is that the endosome looks like basically the same color as the plasma membrane that it originally came from, suggesting that now the luminal leaflet, this is now the internal leaflet of the endosome, has the same approximately similar level of packing as the extracellular leaflet of the plasma membrane. And then we can sort of stain the cytoplasmic leaflet of these same endosomal structures by the microinjection technique and kind of try to infer where those might be. But basically, we don't, when we microinject, we don't see anything close to as red as these colors, suggesting that the biophysical asymmetry of the plasma membrane is maintained quite a long time after endocytosis, and that these endocytic structures are also biophysically asymmetric. Okay, and just for the last bit, a couple more slides on how proteins feel about all this and how they may respond to living in such a biophysically asymmetric environment. So I think it must be important for example, proteins like transporters whose entire role is to change conformation in the membrane spanning regions. But those are a bit hard to get at experimentally. Instead, we just use some bioinformatic tricks to see if proteins can sense this asymmetry of the plasma membrane. And that bioinformatic trick comes from a paper that we recently published, where both experimentally and computationally and bioinformatically, we showed that transmembrane domains can sense the packing of their surrounding lipids, with transmembrane domains that are dissolved in a more ordered membrane tending to be relatively skinny, that is comprised of mostly alanines and glycines, small amino acids that don't have large hairy bits protruding into the membrane and disrupting the packing of the lipids. In contrast, transmembrane domains with, say, phenylalanines or leucines that are kind of have these large hydrophobic regions protruding into the lipids are generally excluded from tightly packed membranes and enriched in these loosely packed membranes. But those experiments and computations were all done in symmetric membranes. And now we know that plasma membrane proteins live in a very asymmetric environment with a tightly packed outer and a loosely packed inner leaflet. So what does that mean for transmembrane domain structure? And to ask that question, we simply looked at all the single-pass transmembrane proteins in the human proteome and compared the total surface area of their outer leaflet-facing part versus the inner leaflet-facing part, and calculated an asymmetry parameter for the total, say, area or shape of the transmembrane domain. That is by dividing the outer versus the inner. So 1, in that case, would be a symmetric transmembrane domain that's sort of a barrel with equal halves, whereas anything above 1 has a larger transmembrane domain area on the outside. Anything below 1 is larger on the inside. And this is what the distribution looks like for all the single-pass plasma membrane proteins now. And what I hope is completely obvious from this bioinformatic analysis is that single-pass TMDs are indeed quite asymmetric, with fatter inner halves and skinnier outer halves that seem to conform to the biophysical lipid asymmetry that we were able to measure in the cells. And that asymmetry that's observable in plasma membrane proteins seems to extend to endosomes and lysosomal single-pass transmembrane proteins, as would be expected from our measurements, whereas the biosynthetic organelles tend to be relatively symmetric, suggesting that maybe their membranes are at least biophysically symmetric. And I don't know what's going on with mitochondria. And finally, that's our analysis for the human proteins. And now we can extend that analysis to really anywhere we would want, because single-pass TMDs are very easy to identify and quantify by bioinformatics. And so here you see mammalian plasma membrane proteins. That's the distribution I showed you earlier. And basically across all clades of eukaryotes, we see a similar kind of asymmetry, with TMDs tending to be skinnier on the outside and fatter on the inside, suggesting that the biophysical asymmetry that we directly measured in these cultured, isolated mammalian cell membranes has been conserved, or rather is maintained throughout eukaryota. So just to quickly summarize the talk, the outer leaflet by lipidomics we find is mostly saturated lipids, whereas the inner leaflet tends to be highly polyunsaturated, and that leads to a biophysical asymmetry within a tightly packed outer leaflet. And proteins seem to have co-evolved, I guess, with this biophysical asymmetry to have a structural asymmetry, and that allows us to infer that the structural asymmetry is present throughout eukaryota. And so I'll just wrap up by thanking the brilliant group of people that I get to work with, in particular Joseph, who did much of that asymmetry work, and Candace, who's my partner in all things life and science. And thanks for your attention, and I'll let Aurélien guide us in the next step.